Morning to everyone. In this lesson, we are going to study about organization of cell and cell organelles. Now, what is present in the cell? Every cell consists of cell organelles. Cell has plasma membrane, nucleus and cytoplasm. All cells are composed of these three components. Cytoplasm is the jelly-like fluid. DNA, which is present in the nucleus, is the genetic material. Plasma membrane. Plasma membrane, this is the outermost covering of the cell that separates the contents of the cell from its external environment. The plasma membrane allows or permits the entry and exit of some materials in and out of the cell. So it is also called selectively permeable membrane. It permits entry of only selected substances. The key concept is the diffusion of substances takes place from the region of higher concentration to the region of low concentration. The solutes move from region of high concentration to low concentration. This is the principle of diffusion and osmosis. Say, carbon dioxide needs to get expelled from the cell. The level of carbon dioxide inside the cell is more and outside the cell is less. So it will diffuse out of the cell. Similarly, oxygen, the amount of concentration of oxygen is more outside the cell. So it will diffuse into the cell. Now, there are three conditions for diffusion or osmosis, which is a passive transport. Passive transport means there is no energy involved in case of passive transports. What will happen if you put an animal cell or a plant cell into a solution of sugar or salt in water? One of the three conditions could happen. It could be an isotonic solution, means the concentration of solute outside and inside the cell are the same. So there is no net movement of water molecules. Remember, it is always the movement of solvent in case of these solutions. Hypertonic. A hypertonic solution has more solutes than a cell. Okay? So what will happen if the medium surrounding the cell has a higher water concentration than the cell, meaning that the outside solution is very dilute, the cell will gain water by osmosis. Such a solution is known as hypotonic solution. Then we have, if the medium has lower concentration of water than the cell, meaning that it is a very concentrated solution, the cell will lose water by osmosis. Such a solution is known as hypertonic solution. So understood what is isotonic, hypertonic and hypotonic solutions? Iso means that there is no net movement of solute and solvent. Hypertonic means they have a lower concentration of water than the cell. And then hypo means higher concentration of water in the cell. So in case of isotonic, there is no net movement. Hypertonic, the cell is going to shrink, and in hypotonic it will expand and it will burst. Then coming to cell wall. The plant cells, in addition to plasma membrane, they have one more layer called the cell wall. Now, composition of cell wall differs from plants, fungi and bacterium. The plant cell consists of cellulose. Functions. The cell wall permits the cells of plants, fungi to withstand hypotonic external media and it withstands bursting. Nucleus is the control center of the cell It is the organelle which controls how much of the genetic material needs to synthesize. It also controls the functioning machinery of the cell. It helps in inheritance of features from parent to next generation. It is the control center, dictates cell in cell division. It plays central role in cell division. It plays a crucial part along with the environment in determining the way the cell will develop. Nucleolus. It is responsible for making ribosomes. Ribosomes are synthesized inside the nucleus and they will be transported out of the cell. Now cells could be of two types, eukaryotic cell and prokaryotic cell. A prokaryotic cell is definitely a bacterial cell. They could be either archaea bacteria or eubacteria. Then eukaryotic cells, we are basically considering plant cell and animal cell. Now coming to cell organelles and their functions. We have endoplasmic reticulum. Endoplasmic reticulum, it is like a tubular network in the cell. Its functions are to synthesize few vesicles. They'll be transporting proteins to whichever region they're required to go. The ribosomes attach to the endoplasmic reticulum, then it forms rough endoplasmic reticulum, and smooth endoplasmic reticulum is when ribosomes are not attached to the endoplasmic reticulum. Functions: smooth endoplasmic reticulum is a site of lipid synthesis and rough endoplasmic reticulum is a site of protein synthesis, and from here they're loaded into the vesicles and they're sent to Golgi apparatus. Now ribosomes are synthesized inside the nucleus and they move out of the nucleus to stick to the endoplasmic reticulum to form proteins, or they may either move in the cytoplasm. Golgi apparatus, it is like a factory of the cell which is modifying the proteins from the endoplasmic reticulum, which come from the endoplasmic reticulum, and they are sending it to the place where they are required. It was discovered by Camillo Golgi. Functions of Golgi apparatus: formation of complex sugars from simple sugars, formation of lysosomes. Now lysosomes, what are lysosomes? Lysosomes are the suicide bags. They are loaded with digestive enzymes, and when they come in contact with something which is infecting your body, they burst open and the digestive juices are poured over the microbes and they lose their life. So they are known as suicide bags. Mitochondria is the powerhouse of the cell. It is the cell where ATPs are synthesized. Plastids could be chloroplasts, leucoplasts and chromoplasts. In addition to the cell, the cell has a nucleus. These plastids are autonomous. This means that they also have their own DNA and ribosomes, so they are semi-autonomous. Like they can synthesize on their own. They can multiply on their own whenever they require. Chromoplasts, leucoplasts, and chloroplasts. The functions of plastid. The most important plastid is chloroplast, which is very important for photosynthesis. Leucoplasts are used for storage, and chromoplasts give color to the flowers and fruits. Vacuoles. In case of plant cells, vacuoles are larger in size. In case of animals, they are very smaller in size. These are the organelles which help in storage of additional food substances. Now, can you identify the cell organelles here? Hope this video was useful.